Hello everyone. In this class, we'll try to learn about neuromuscular junction. It is the junction between the motor nerve and skeletal muscle fiber. Nerve membrane, which is in close approximation with the muscle membrane, is called presynaptic membrane, while the muscle membrane is called postsynaptic membrane. The space between these membrane is called as synaptic cleft, and it measures around 50 to 100 nanometers, which is filled with extracellular fluid. Postsynaptic membrane has many folds of muscle membrane called subneural cleft, which increases the surface area for the synaptic transmitter. Axon terminal has many mitochondria, that is for the synthesis of acetylcholine, and minute vesicles or granules which contain small packets of chemical transmitter acetylcholine, which is responsible for synaptic transmission. An enzyme specific acetylcholinesterase is found in high concentration in postsynaptic clefts, which destroys acetylcholine. Synthesis, storage and release of acetylcholine. Acetylcholine is synthesized within the mitochondria from choline in the presence of enzyme choline acetyl transferase, or also called as choline acetylase. In addition, it requires acetyl coenzyme A, ATP and glucose. With the exception of choline, all the components required for synthesis of acetylcholine are present within the mitochondria, or they gain ready access to them. Acetylcholine, once formed, is temporarily stored in the minute vesicles, which are called as synaptic vesicles. The nerve impulse causes fusion of vesicles with the membrane of terminal nerve fiber by increasing the permeability of the calcium. Thus, released calcium from the ECF helps in the fusion of changing ionic composition and thereby decreasing the flow of exoplasm. Therefore, vesicles easily come in contact with the membrane, and magnesium decreases this process. The amount of neurotransmitter released is directly proportional to the calcium influx. So this is the diagram showing neuromuscular junction. So this is called as presynaptic membrane, and this is called as postsynaptic membrane, the membrane of the muscle or sarcolemma. So when an impulse, and the presynaptic membrane contains many structures like mitochondria, which is important for synthesis of neurotransmitter acetylcholine, which are stored in small vesicles. When action potential comes to the presynaptic membrane, it increases the permeability of the calcium by affecting the voltage gated calcium channels. When calcium enters inside, it binds to the vesicles, and the vesicles come closer to the presynaptic membrane, and there they are going to release the neurotransmitter acetylcholine. Postsynaptic membrane has receptor for this acetylcholine, and when acetylcholine binds to these postsynaptic receptor, they helps in the opening of the ligand gated sodium channels, so which is going to responsible for postsynaptic potential or end plate potential. The final step is destruction of acetylcholine within 1 milliseconds after acetylcholine is released by acetylcholinesterase. It appears that vesicle breaks with the membrane when VAMP, a vesicle membrane protein, connects with the syntaxin, a cell membrane protein. The connection occurs in the presence of G proteins like alpha SNAP, beta SNAP, SNAP25 and NSF. So this diagram showing the transmission at the neuromuscular junction. So when at presynaptic membrane at the axon terminal, if the action potential arises, it opens the calcium channels, which causes entry of calcium ions from the ECF into the presynaptic membrane, which causes the opening of the vesicles and release of acetylcholine into the synaptic cleft. Then the acetylcholine is going to bind with a receptor, and there will be formation of acetylcholine receptor complex, which open the ligand gates of the sodium channels. Thus, sodium entry occurs from the extracellular fluid into postsynaptic membrane, that helps in the development of the end plate potential. And this end plate potential added and it leads to generation of the action potential not in the postsynaptic membrane but the sarcolemma of the muscle. The toxins which blocks the neurotransmitter release. D-tubocurarine competes with acetylcholine for the same nicotinic receptors on the motor end plate, whereas tetanus and botulinum neurotoxin B cleaves VAMP, whereas botulinum neurotoxin B cleaves SNAP 25. Note that tetanus causes spastic paralysis and botulinum causes flaccid paralysis. The drugs which stimulate neuromuscular junction by inactivating acetylcholine esterases are neostigmine, physostigmine, and diisopropyl fluorophosphate. Myasthenia gravis. It is an autoimmune disease in which patients have developed antibodies against their own acetylcholine activated ion channels, that is nicotinic receptors. These antibodies destroy acetylcholine receptors, that is nicotinic receptors, thereby causing muscle paralysis due to the inability of the neuromuscular junction to transmit enough signals from the nerve fibers to the muscle fibers. The disease is characterized by a rapid onset of fatigue with marked generalized weakness of the muscles. The most commonly affected muscle are extraocular muscles, facial, the swallowing is affected, and the muscles of mastications are affected. In severe form, the patient may die with a paralysis of respiratory muscles. Treatment. In myasthenia gravis, which is an autoimmune disorder, antibodies are produced against nicotinic neuromuscular receptors in the neuromuscular junction. To prevent the action of the antibody, we have to increase the release of the acetylcholine. For that, the cholinesterase enzyme has to be inhibited by anticholine esterases such as neostigmine. Lambert-Eaton syndrome. In this, muscle weakness is caused by antibodies against one of the calcium channels in the nerve endings at the neuromuscular junctions. This decreases the normal calcium influx that causes acetylcholine release and it leads to paralysis. This is in brief about neuromuscular junction. Thank you.